Greetings, everyone. Welcome to a guide to understanding basic concepts in bioinformatics. My name is Tracy Marin, and I will be your speaker for tonight. A disclaimer, all content used on this presentation is owned or licensed by Crimson Interactive Incorporated or its affiliates under the CC BY NC SA 4.0 license. Some content may have been blocked for confidentiality reasons. Unauthorized use of any part of this presentation by any other party is prohibited. Breach of this condition is liable for legal action. So to get started, a quote from J.D. Tisdall from the beginning of Perl for bioinformatics in 2003. If you can't do bioinformatics, you can't do biology. So this was more than 10 years ago when we started to realize how important bioinformatics was to the growing field of biology. So our agenda for this presentation tonight is a basic overview of what bioinformatics is, describing the essential biological concepts that we need to understand for bioinformatics, some insights to biological databases that are available online, and how to access these online resources for your bioinformatics analysis. Starting with point 1 or agenda 1, an overview of bioinformatics. So what is bioinformatics? It is an interdisciplinary and highly collaborative field, including disciplines such as biology, computer science, statistics, mathematics. All of these work together to give us a broader view of what's happening at the biological level. It involves a lot of expertise, such as biological programming, software development in which we can use that biological programming, hardware development that allows us to use the software that we've developed, and of course we need the experimental design and the experiments. All of this put together with some other things also is what we call bioinformatics. So what's the purpose of bioinformatics? Simply, it is the use of computational tools to take large databases that are complex that we were able to derive from biological experiments, use information technology to make sense out of it, to give us clues, to increase our biological knowledge. So it's basically using programs to analyze complex biological data to drive our hypotheses and understand what's happening in these large databases and the biological organisms. So some basic applications of bioinformatics, genomics. Genomics is where we kind of start. Bioinformatics manages the large amount of data generated from gene sequences, their interrelation and functions by providing both theoretical basis and practical methods for analyzing systemic functional behaviors of the cell and the organism. And then moving up the hierarchy, proteomics. Proteomics results or protein data generated from techniques such as peptide mass fingerprinting and peptide fragmentation fingerprinting, gel technologies, HPLC, and mass spectrometry are managed through several bioinformatic databases, tools, and softwares. And of course, we want to look at the transcriptome level also. So bioinformatics can be used to determine mRNA levels, their expression profiles through microRNA techniques, which involve numerous experimental runs resulting in the generation of a large amount of data. Bioinformatics is widely used for analyzing these transcriptome data. I've used this a lot in my research, actually. Bioinformatics also facilitates chemoinformatics analysis, similarity searching, clustering, QSAR modeling, etc. This is performed for developing a new compound with the desired properties and assessing its therapeutic effects. That leads us to drug discovery and drug development. So bioinformatics and computational methods have become essential for drug discovery and drug development. We basically don't start any of this without bioinformatics nowadays. Clinical, preclinical findings are analyzed and interpreted to predict drug likeness by the means of bioinformatic tools. And then, of course, we can take that and the applications we talked about in the previous slide to move it into other applications such as personalized medicine, agriculture science, veterinary science, forensic science, evolutionary biology, biodefense, biofuels, and bioenergy, and of course, much, much more. We should add public health into this. The scope of application for bioinformatics, this gives you kind of a basic idea. It expands from this, however, but using bioinformatic analysis, we can predict nucleic acid structure, predict protein structure, identify protein structure classifications, and compare different protein structures. At the genome level, we can compare genomes of one organism to another or one person to another. We can look at genes and their promoters and what might be regulating those promoters. We can discover different motifs in the genome that help us understand gene regulation. And of course, we can sequence databases and searching for what's up or down in particular data sets and sequence alignment. From a functional perspective, we can model metabolic pathways and look at how gene expression profiles might play a role in regulating pathways and networks. We can look at protein interaction and protein interaction and network nodes. We can also look at where proteins might be in the cell, so protein subcellular localization prediction and whatnot. Using bioinformatics, we can basically predict or create algorithms that allow us to see what's happening inside the cell at the molecular level. So what's the difference between bioinformatics and computational biology? Well, bioinformatics involves the use of computational tools and systems to answer problems of biology. Computational biology involves developing the theories, algorithms, and techniques for bioinformatics, for such tools and systems. So they complement each other, but we need computational biology to implement bioinformatics. So moving on to understand the essential biological concepts that we need to understand for bioinformatics. It revolves around three basic biological processes, and most of you are probably familiar with this. Number one, we have the DNA sequence, which is going to determine our protein sequence. Our protein sequence is going to determine the protein structure, and of course that protein structure will determine the function of that protein. Is it an enzyme? Is it a transcription factor? Is it a chaperone, etc.? And of course my favorite, the basics of the central dogma. Obviously it's a lot more complicated than this because we have to consider epigenetics now. But the idea of the central dogma starts with the DNA. The DNA is transcribed by RNA polymerases to produce mRNA. That mRNA then is going to be translated into peptide chains, and these peptide chains will be combined to produce polypeptide chains, which ultimately will result in our protein, the synthesis of protein. So we like to look at every level of the central dogma using bioinformatics because it allows us to understand gene regulation and gene expression and how those genes are regulated relative to the environment and what those genes might be doing in different networks. This is just a little diagram reminding us of how that gene actually produces a protein. So if you look to the left side, we have the gene, exons, intron, exon. And as you know, it's the exon that's going to be transcribed into the mRNA. And the mRNA will be comprised of three nucleic acids that will nucleotides, that will be translated into a particular amino acid using tRNA. And as those amino acids are produced, they will be combined to produce a polypeptide chain. Those polypeptide chains will be bound to other polypeptide chains to ultimately produce our proteins. Now, reading frames. Reading frames can be a little complicated. But looking at this diagram, let's think about a reading frame as a way of dividing the sequence of nucleotides in a nucleic acid, such as DNA that you see here, in a DNA molecule, into a set of consecutive non-overlapping triplets, where these triplets will equate to an amino acid or stop signals during translation. Each of these triplets is called a codon. A single strand of nucleic acid molecule has a phosphoryl end, called the 5' end, and a hydroxyl end, or the 3' end. These define that 5' to 3' direction. We've all learned this before. There are 3 reading frames that can be read in this 5', 3' direction, each beginning from a different nucleotide triplet. In a double-stranded nucleic acid, such as this, DNA, an additional 3 reading frames may be read from the other complementary strand in the 5' to 3' direction along this strand. As the 2 strands of a double-stranded nucleic acid molecule are anti-parallel, the 5' to 3' direction on the second strand corresponds to the 3' to 5' direction along the first. So basically, a double-stranded piece of DNA can thus have a total of 6 different frames of reference for demarcating codons. Each of these 6 is called a reading frame. The genetic code and using the amino acid codon wheel. This is a useful tool to find which amino acid is translated from an RNA sequence. The codon wheel is used by scientists, researchers, and students during RNA translation to find the amino acids for that sequence as a quick, easy reference tool. I would have liked to be able to use this when I was taking my exams in PhD school. So how do we use this wheel? To use an amino acid codon wheel, start from the center. So the center here, we have A, G, C, U. Start from the center and follow the RNA codons until you have the 3 nucleotide bases. So if I wanted to start with C, and then G, and then C again, we have arginine. So this process is what we call RNA translation. Once established, follow the RNA sequence to find the amino acid that it translates to. The protein that the mRNA translates to is made up of those amino acids. And of course, all of that results in the building blocks of proteins, amino acids. So what are amino acids? Remember that each protein consists of 1 or more polypeptide chains. Each polypeptide chain consists of smaller subunits or amino acids that are linked together. It's these amino acids that serve as the building blocks of polypeptides, and polypeptides serve as the building blocks of proteins. As you can see here, there are 20 types of amino acids commonly found in proteins. Amino acids share a basic structure which consists of a central carbon, also known as the alpha carbon, bonded to an amino group, NH2, a carbonyl group, COO, or H, and a hydrogen atom. So what can we learn from using bioinformatics to analyze big data, high throughput screening, big data? Well, a lot. A lot can be predicted on the basis of genome sequence. You can determine the identity of an organism. You can predict the structure of proteins. You can identify protein families and domains on the basis of homology. You can predict metabolic pathways. You can predict metabolic networks, and the list goes on. So what are databases? Well, they're computerized archives that provide easy access to data through various search criteria. Data management or database management involves computer hardware and software, as we mentioned early on. Databases aim to organize data in the form of structured records to enable ease of access to users. In addition to the above-mentioned objectives, biological databases aim to serve as knowledge discovery platforms, enabling the identification of connections between related information that was not initially known. So, biological databases and their classification. There are primary databases, And these primary databases contain nucleic acid or protein sequence data, such as ENA or GeneBank. And then we have secondary databases, which contain the results from analysis of data in these primary databases, such as ProCite, PFAM, blocks, etc. Now, within those databases, we like to look for nucleic acid databases or protein databases. And nowadays, there's a lot more databases also that include methylation of CpG units, acetylation of histone, etc. So the databases are growing and growing. But here, for simplicity, we're just talking about nucleic acid databases and protein databases. So over here is an example, are examples of databases that contain nucleic acid information. So for example, we have EMBL, GeneBank, and DDBJ. Remember DDBJ for later. Now, I have tried to click on these to go through the process of showing you these databases online, but the system is not letting me. But just consider clicking on GeneBank when we're finished, and from GeneBank, you can go into another database that's called Intras Nucleotide. In this link, you can identify and annotate different genes or recognize genes and their identifiers and annotations. You can also use BLAST, B-L-A-S-T, in GeneBank to do sequence queries or alignments. You can also search, link, and download sequences using NCBI eUtilities, which is also found under GeneBank. So let's talk about some online resources for bioinformatics analysis. NCBI, I think most of us are familiar with NCBI, the National Center for Biotechnology Information. This is a resource center for tools and databases related to bioinformatics, among many other things. There's also the European Bioinformatics Institute. From this, you can conduct research and find services that are related to bioinformatics. And this website manages databases of big biological data. There's Ensemble, which we mentioned a little bit earlier, which will allow automatic annotation of databases. XPASI, providing access to scientific databases and software tools in different areas of life sciences. I encourage you to click on all of these later and explore them. Continuing our talk about NCBI, NCBI provides a plethora of information in the fields of medicine and biological sciences. You can use it for database, books, publications, etc. It hosts several bioinformatic databases and analytical tools. A few from the several featured of NCBI website are highlighted in the next two slides. So here we have the 1000 Genomes browser, BLAST, ClinVar, eUtilities, and Gene. All of these, in some way or another, allow you to graphically view, explore, align, look for sequences, annotate, and compare different databases related to the genome and proteomics. We also have the Gene Expression Omnibus. A lot of people use this to annotate their databases. The Genetic Testing Registry, where you can look for information on genetic tests in laboratories. MedGene, which organizes information related to human medical genetics. The Nucleotide Database, which is a collection of sequences from several sources, including GeneBank, RefSeq, TPA, etc. RefSeq, excuse me. The Sequence Read Archive is NIH's primary archive of high-throughput sequencing data from a variety of sequence platforms. And continuing on to the European Bioinformatics Institute, EBI. EBI makes the world's public biological data freely available to the scientific community via a range of services and tools. They lead computational biology research with work spanning sequence analysis methods, multi-dimensional statistical analysis, and data-driven biological discovery, from plant biology to mammalian development and disease. They host a series of training events and online courses to train scientists at all levels to get the most out of publicly available biological data. Some examples of tools that are available by EBI: Clustal Omega, which allows for multiple sequence alignments of DNA or protein sequences. Interprocan, which allows you to search sequences against its predictive protein sequences. BenNICH, which is a chemical class enrichment analysis database. HMMER is fast sensitive protein homology searches. It allows you to search sensitive protein homology using profile hidden Markov models, HMMs. Assembly Converter, which allows you to map your data to current assemblies. And Bupatist, which is a tool to analyze two ontologies, typically two versions of the same ontology. And here are some more. ArrayExpress, a database of functional genomics experiments, including microRNA and RNA-seq. Biomodels, which is a repository of peer-reviewed published computational models. Biosamples, a database describing biological samples and providing links to associated experimental data. This allows you to look at that data yourself. GEBI, reference for chemical structures, nomenclature, and ontological classification. CHEMBL, ChemEMBL, an open data resource of binding, functional, and admin bioactivity data. Complex Portal, a manual curated encyclopedic resource of macromolecular complexes from model organisms. And DBFetch, a programmatic style retrieval of entries from EMBL, EMBI databases. Ensemble, which I've used a lot, is a genome browser for vertebrate genomes that supports research in comparative genomics, evolution, sequence variation, and transcriptional regulation. Through Ensemble, researchers can annotate genes, they can compute multiple alignments, predict regulatory function, and collect disease data. These tools include BLAST, BLAT, BioMart, and the Variant Effect Predictor, VEP, for all supported species. Key features of Ensemble: compare genes across species, find SNPs, single nucleotide polymorphisms, and other variants for your gene of interest. Look for gene expression in different types of tissue. Retrieve gene sequences, find a data display, or use your own data in Ensemble. XPASI is a bioinformatics resource portal which provides access to scientific databases and software tools for areas of life sciences such as proteomics, genomics, evolutionary biology, transcriptomics, biophysics, imaging, glycomics, etc. Very useful. Okay, so that is the end of our bioinformatics presentation.